Our next speaker is Dr. Sam Sinclair from BYU. He's going to be talking about the physiological ecology of Aspen-Conifer interactions. Sam is an assistant professor there at Brigham Young, and I think arrived 2 years ago. His education is from BYU and Penn State University. Did postdocs at Stanford and the University of California at Berkeley. His research interests are in plant and ecosystem responses to abiotic and biotic stresses in the context of resource limitations. Current projects are Aspen ecophysiology and Mojave Desert research. Okay. Well, anyway, he's going to explain that all to you.
Yeah, I don't write very much anymore, mostly on the keyboard, so sorry about that, Dale. Talks that have already been given lead in perfectly with what I want to talk about, so I want to thank the previous speakers. And I will talk about repeated defoliation events, some biotic, some abiotic today. So what is physiological ecology, first of all? We've talked about climate, we've talked about insects. I'm going to take you and try to explain what the aspen are experiencing, what are they sensing, how are they responding to these changing conditions. So an ecophysiologist or a plant physiological ecologist will often use this framework to understand ecological observations. You have a potential species pool, and there are three filters. First, there's a historical filter, and you can ask the question, does it arrive? Aspen is really interesting because it has arrived in a lot of places, from the Atlantic to the Pacific, all the way up to northern Alaska, down to central Mexico. So you have to ask, what are the traits that have allowed aspen to be so successful across that wide amplitude of environmental gradients? The second is a physiological filter. Can it germinate? Can it grow? Can it survive? Can it deal with the climate conditions to which it's exposed and successfully reproduce? And the third filter is a biotic filter. Does it successfully compete and defend itself under the conditions in which it finds itself? So we generated this conceptual model in my lab to look at these different factors that may contribute to the success of aspen, or more recently, some of the observed decline, dieback events. And I really become interested in extreme climatic events, which are on the rise based on the models and predictions. Specifically looking at frost damage events and then extreme drought events. And the idea being that these extreme climatic events lead to carbohydrate depletion, which is the resource that plants use to better compete against competitors and to defend itself against herbivore pressures. So two general things can happen with extreme drought events and repeated freeze-thaw cycles. You get reduction in photosynthesis, you get leaf area loss in the canopy, and you can also get cavitation. And so in the xylem cells that transport water, you get expansion of air bubbles that prevents water transport. So the plant, or aspen, then have a dilemma. Okay, in the background of carbohydrate depletion with extreme climatic events, are you going to be a better competitor and grow with the resources that are available, or are you going to allocate resources to defense chemistry to defend yourself? So the first study question I want to go over is how does extreme climatic events influence aspen physiology? This is July 2nd, so almost to the 4th of July. You can see the understory is very green. This is an aspen clone in the Uinta forest, no leaves. They should have their leaves by this time, okay, into May, mid-May. So what happened is in 2007 we experienced an early spring that accelerated leaf flushing in the aspen. And May 23rd, temperatures plummeted at high elevations, and we had this widespread aspen defoliation. This is the Fishlake. Again, this is late June, early July, and just complete defoliation of the aspen. And this is an abiotic defoliation in response to the frost damage event. So
they didn't leak back out?
I'll get to that. They did, yes. So the first question we had is, how widespread was this defoliation? So we collected some MODIS satellite imagery and tracked NDVI, which is a measure of canopy greenness. So from satellites, we're looking at changes in greenness over the state of Utah. And we identified these aspen-rich polygons and overlaid these polygons where there's a lot of aspen over the NDVI images. Okay? And these polygons were defined based on the Southwest Regional GAP database. And when we looked, this is the solid line is the 2000 to 2004 mean NDVI over these 37 polygons. And this dotted line is 2007. Okay? And indeed, we did see early greening right here in these aspen-rich polygons. And then this blue area represents the period in which the frost damage occurred. And we didn't see a drastic drop in canopy greenness, but we did see a reduction in the slope relative to the 2000 to 2004 mean. And when we charted this out spatially over the polygons, what we found, these orange regions are areas where there was a significant decrease in canopy greenness. And frost damage is most intense in more northern polygons at intermediate elevations. So yes, they did leaf back out. This is May 31st. This is at Deseret Ranch. We came back in the middle of July, and the aspen had leafed back out. And this physiologically is very interesting because you've lost the resources as these leaves became necrotic and fell off. And so there's a resource investment that has to then go back in to reflush it. The question we were really interested, though, in was, is there induction to defense chemistry? Of course, insect herbivores defoliate aspen, and Rick Lindroth in his lab has shown that you get induction to defense chemistry following a biotic defoliation event. We were wondering, does the same thing happen with an abiotic defoliation event? So we had two study sites. Deseret Ranch, we had a transect study along a ridge where there was severe frost damage, and then down in the Fishlake National Forest on South Last Chance. So here's a transect here. We had three classes. Okay? We had clones that experienced no frost damage. They retained their first flush leaves. And we had an intermediate frost damage class where there was complete defoliation, and a second flush that was complete. Okay? It reflushed completely. And then there was an extreme or severe frost damage class where there was complete defoliation but only partial reflushing. And the morphology and the leaf of physiology is actually very interesting, but I don't really have time to go over that. Here you can see the necrotic leaves. It was very easy to spot the trees that had lost their first leaf flush. And then you can see the reflushing here. We measured a bunch of different physiological parameters to see how physiologically the aspen had responded to this frost damage event. On the Fish Lake, we identified a site while we were out with Bob Campbell, where one side of the aspen tree had lost its first flush, but retained its first flush on the other side. And we came back, and this had reflushed, so we knew these were second flush leaves, and these were first flush leaves on this side. And we did the same type of analysis, but on the same tree, the same clone. And what we saw across the board, here we have condensed tannins on the top, phenolic glycosides in both studies. We have the control on the left, the intermediate severe frost damage. With the exception of condensed tannins in the Deseret study, the severe frost damage class, we see 2 to 3-fold induction of defense chemistry. Okay? And what's interesting and important about this is look how much the resources that are being allocated towards defense. The units here are percent dry weight. So we're talking between 15, 20, 25, maybe as high as 30% of leaf dry weight can be allocated to produce these defense compounds. Okay? So when the plant makes a decision to allocate these resources like this to defense, they're simultaneously making the decision not to allocate those resources to growth. So what's the idea behind this? Why would you induce drastically your defense responses following an abiotic defoliation event? And one hypothesis we have is the insurance policy hypothesis. And Jesse talked about the potential for the gypsy moths, the forest tent caterpillars are here. But what we see, and when we look at the leaf nutrients in first flush leaves versus second flush leaves, we see about a 30 to 35% reduction in many of the major macronutrients as you go from a first flush to a second flush. I will tell you, photosynthesis does not change. Okay? There's no reduction in photosynthesis, but the idea is if this abiotic defoliation is followed by a biotic defoliation, your nutrient thresholds could be dropped below a threshold point that would then really impair physiology. The other idea is they just can't distinguish between abiotic and biotic defoliation events. And so defoliation itself is a signal that you need to induce defense responses. And so they may inappropriately be allocating resources to defense when there's really no threat. In fact, if you had an extreme frost event, your insect loads may be less. So now I want to talk a little bit about drought. This is a part of the project we're just starting up. I made a trip down to northern Arizona. This is Mary Lou Fairweather, who kindly took me around. We're interested in the influence of drought and drought-induced cavitation, or the ability to transport water up to the top of the tree on this dieback phenomenon we're seeing in some areas, particularly in Colorado and northern Arizona. And if you look at the pattern, what you'll often see is that the dieback occurs at the top. And this would be consistent with an inability to transport resources because your xylem cells are cavitated. And so we identified a couple of low elevation sites that have drier conditions and a couple of higher elevation sites. And we have just started to collect and analyze this data, and I'll show you what I'm doing there. This is actually a study I did in 2004 with Ron Ryle in Green Canyon, which is here in Logan. And this is a common garden. And we looked at aspen growth responses to 10 years of drought. And this is precipitation data that was collected at a station nearby. And what's interesting about this is vertical growth of the 20 clones in the aspen garden over the period of 1984 to 2005. And diameter growth, or girth growth of the stem is maintained over the period of the study. What's interesting is you actually get a height plateau from 92 to 2005, which corresponds to some pretty dry years. And again, that supports the idea that the cavitation may be playing a role in inhibiting the transport of those resources to the top of the tree. And so the top of the tree dies out, and you don't get continued vertical growth. What's interesting is there is some clonal variation. Okay? You know, a lot of them flatten out, but there are a few clones that are able to maintain not a consistent growth rate, but some positive growth. And so we're interested in the physiological traits that allow that continuation of vertical growth in those clones. And here's some photosynthesis data we collected in the summer of 2004, which is a very dry summer. And if you look at photosynthesis among the 20 clones over the course of the summer, you get a decline in stomatal opening, conductance that then impairs the ability of the leaves to photosynthesize. And what was interesting is the winter 2004-2005 was actually a pretty good winter. And so we believe recharged the soil moisture. And so here is this data right here, just transferred over here. And then if we look at how these clones at the same time point the next year, when water relations are better, you actually see a pretty consistent increase in photosynthesis when moisture conditions are better. The really interesting trend is a lot of these clones that had the lowest rates of photosynthesis in 2004 had some of the highest rates in 2005. And the idea is that one way you can control cavitation is to close your stomates. That limits the tension placed on the xylem cells that causes cavitation. So we think these clones that have low rates of photosynthesis are limiting the cavitation that may be occurring, and that shows up as low rates of photosynthesis under dry conditions. But then they're not cavitated when there's greater water availability, and they can have much higher rates of photosynthesis. And I've been working in John Sperry's lab at the University of Utah this summer trying to get some hydraulic trait analysis done. So I went and collected these aspen stems down with Mary Lou in northern Arizona. And again, these are the dry sites, and these are the higher elevation, wetter sites. It was actually a pretty good water year down there. So not a good year to actually maybe do this study, but maybe gives us a good baseline when we have a drier year to compare against. So what we do is we take these stems and we put them into the system. You have a pressure head here, and you can force water through the stem, and then the water will come and you can measure the amount of water that's flowing through based on the increase in mass on the balance. And then what we do is we flush all the cavitated air bubbles out of the stems, and we remeasure, and from that we can calculate percent cavitation. Okay? And these are not in their final units, so I hesitate to even show you this data, but the general trend is that these lower elevation, drier sites tend to be more cavitated than the higher elevation sites. But the actual native cavitation for this year was pretty good, which is evidence that it was a good moisture year down in northern Arizona. So we're going to continue this work and try to understand how hydraulic traits in aspen clones contribute to their success or maybe their failure based on drought conditions and freeze-thaw events. So back to our model. What we do show is these frost events do lead to massive leaf loss. And there is carbon that's lost, so the potential for carbohydrate depletion is there. Cavitation, I do think, plays a major role. So I think we're looking, under these conditions of extreme climatic events, less carbohydrates to work with as you go into this dilemma right here. I need to defend myself against ungulate pressure, herbivore pressure, pathogens. But the other problem is, I would say, and I think most of you would probably agree, one of the major contributors to aspen decline is actually succession to conifer in the face of fire suppression. And I just showed you that in response to defoliation, the aspen has made a decision to allocate resources to defense. Okay? And by doing that, there's less resource there to actually defend yourself against conifers that are good competitors and generally more drought tolerant. So follow the study questions that I want to address in this section. What are the mechanisms underlying aspen conifer succession? Does aspen facilitate conifer establishment? This is the first question I want to get at. And then how do shifts in overstory composition influence regeneration success? So we can look at a forest right now, and this is the present, but underneath the regeneration that's occurring and the dynamics in the understory will define what the future forest looks like. Okay? I'm really interested in how the overstory composition influences the regeneration success. And then one pattern that is really interesting is the aspen really do seem to facilitate the establishment of the conifer. Right? So we'll go along, you'll see these beautiful forests, and then they'll just stop at this boundary. And the conifers will stop where the aspens stop. So what's going on in terms of these establishment patterns? We're very interested in that. And so we began doing surveys, and the initial observation was that the aspen boundary defined the conifer boundary. But as we looked more closely, we actually found a facilitation effect. Okay? You'll see here's an aspen, and where we were finding the subalpine firs establishing at a finer spatial scale was actually right next to these aspen trees. Okay? And just be a good observer. Good science is initially about good observation, right? So you can see these subalpine firs getting bigger, and it's established right next to this aspen. If you look at the interspaces, you find very few subalpine fir. Where you see them establishing is right next to a mature aspen tree. Same kind of thing. And eventually they get bigger and bigger. Same kind of thing. The interspaces, you just don't see much subalpine fir establishment. You always find them right next to an aspen. And eventually it reaches this stage where they begin overtopping the aspen, competing for resources. Light resources in the above canopy and water and nutrients below ground. So we designed a study, 7 sites on the Fishlake National Forest in 7 transition zones. And here's a transition zone right here, where on the left we have relatively pure conifer. We have a mixed aspen conifer component, and then more of a pure aspen stand, and then we also looked at gap dynamics. And we use point quarter transects and nearest neighbor estimates to determine overstory composition and the spatial relationships between overstory trees and regeneration. And for these 7 sites, what we found is our relatively pure conifer stands were about 86% to 14% aspen. The mix was a 47% conifer, 53% aspen. And then the more pure aspen was about 7% conifer, 93% aspen. The gaps didn't have anything. And this is what we found in terms of establishment patterns. Okay, so this is subalpine fir seedling that's equal or less than 5 centimeters from an overstory tree, okay, in each of the transition zones. Okay, the pure, the mixed, and then the pure again. And if we look at overstory aspen in each of these different zones, between 37 and 50% of the aspen trees had a subalpine fir establishing right next to it. And when we looked at the subalpine fir, it just wasn't the case. Very rarely did you find that was the case. And we're still doing the analysis, but we've done quadrat studies, and again, the interspaces, there's just not a lot of establishment. So with my plant biology class, I had them come up with a list of ideas they had that could explain this facilitation effect. And then here's a list that they came up with. I didn't include everything. I asked them to be creative, and they got really creative. Less understory competition. You can see these little spaces where there's a root extension. These interspaces are highly competitive with their herbaceous community. Right? So you can get more light if you're cuddled up next to an aspen. A stem flow. Right? So you get precipitation events, stem flow, nutrient concentration. It gives you better water dynamics right around the aspen tree. Seed dispersal. Both animal influence and also, again, wind maybe hitting the branch and maybe falling down and concentrating seeds closer to the aspen. This just kind of popped. Snow tree wells was another idea. Herbivory deterrence. Sometimes herbivores, they like to be able to see what's going on, especially if they're sensitive to predators. And if you're blocking your view in one direction, then that may be a problem. I doubt that's what's going on, but they were being creative, and I thought I'd put it on the list. So another interesting question is, and it's not like even aspen had this choice maybe, but why would they facilitate the establishment of their competitor? And one idea we kind of come up with is pure aspen stands do not burn. So if you facilitate the establishment of a mixed stand, then you can burn, and we know that aspen can then, with great vigor, regenerate. The problem is if you suppress fires, then that regeneration doesn't occur with the burning event, and then that's when you get the overtopping and more of a competitive situation. So we then wanted to kind of tie in some of the ecology with some physiology. Here's my graduate student, John, and we went along these transects and randomly located aspen regeneration and collected leaves. And we looked at the effect of overstory composition on phenolic glycoside production. Phenolic glycosides are defense compounds that are really efficient at deterring herbivory. And what we found is that there's a lot more phenolic glycoside production in gap and pure aspen stands than in mixed or conifer stands. Okay? The question is, what's the driver? And here's a boundary between a pure aspen stand transitioning into a mixed stand. And there really are a couple of things that could be going on. Of course, the light environment is very different. There's more light that reaches the understory of an aspen. There's more of an herbaceous community. The other thing that could be going on is soil engineering. Okay? So as the conifers establish, they'll change the soil chemistry, the soil microbial community, and so it could be some kind of a below-ground resource influence as the mixed stand changes the composition of the soil. So we did a controlled study to look at this. We collected soil cores underneath pure aspen stands and then in mixed aspen conifer stands. And we came back and conducted a greenhouse study where we manipulated light conditions, and based on our soil cores, where we collected them from, we had aspen soil cores and conifer soil cores. This shade cloth is representative of what you would get in a pure aspen stand, and then the additional shade boxes are more representative of light environment in a mixed aspen conifer stand. And then we look at the growth and physiology of aspen and subalpine fir growing on these soil cores under these different light environments. And it's a complete factorial split plot design replicated 4 times. And here are light response curves. So this is photosynthesis with increasing light intensity as a function of light environment and soil conditions. And this is just for aspen. And what you see is that under low light environments, under the shade boxes, aspen photosynthesis is not influenced by soil type. But when you get out in a condition with greater light intensity, the aspen much prefers being on self soil as opposed to conifer-influenced soil. So your rates of photosynthesis are much higher under a high light environment on aspen soil. And here are the growth responses. And again, this is aspen, subalpine fir under high light environments or low light environment. And then the black boxes are seedlings, or we'll call them seedlings, growing on aspen soil or conifer soil. And you see that light environment is important for both. So seedlings growing under low light environments have much lower growth, but it's more, I would say aspen's more sensitive than the subalpine fir. Soil has no influence on subalpine fir. Okay? Regardless of light environment, when it's growing on aspen soil or conifer soil, there's no growth effect. For aspen, that's not true. Okay? Especially under high light environments, they're much more sensitive to the conifer soil. They grow much less than on self soil. And to get at a little bit of a mechanism, why is subalpine fir more tolerant of conifer soil? One thing they seem to be able to do is on conifer soil, it seems to adjust the root to shoot ratio. So they allocate more resources to root growth, and they capture nutrients that are more limiting in the conifer soil. Okay, so here's the soil chemistry data. Here's aspen soil, conifer soil. pH isn't much different. Bulk density isn't very different, but there's a huge difference in nitrogen. There's more nitrogen in the aspen soil. Actually, more phosphorus in the conifer soil, and much higher concentrations of base cations in the aspen soil. So if you want to capture these limiting nutrients, then you can allocate more growth towards roots, and these higher values represent more allocation to root growth. And so it looks like the conifers, the subalpine fir, are allocating more of their resources to root growth to be able to capture the limitations of nitrogen and maybe base cations. Phenolic glycosides, I don't have the tannin data yet. I cranked out this graph last night at about 11:30. So this is data that's hot off the press. My undergraduate students, my graduate students are doing a great job this week in getting this data together. So going back to what is driving the defense response, okay, and I told you it could be the soil differences, or it could be light environment differences, and it looks like light environment's driving the differences in at least phenolic glycosides. Okay. So here we have the aspen soil and the conifer soil. Black bars are the high light and the low light environment. This is aspen defense chemistry. And what you see is there are no big differences in phenolic glycoside concentrations when we compare them growing on aspen and conifer soils. But when you go from high light to low light environment, you get a drastic reduction in phenolic glycosides. And so thinking back on our conceptual model, the influence of the aspen-conifer interaction is having an influence on physiology and defense responses that may be really important. So this is my thank you slide. I really want to put in the acknowledgement section of one of my papers a thank you to Gus, my 1993 GeoPrism. I've taken this car everywhere and it's never failed me. It gets good gas mileage, and I'm a young assistant professor, so I'm trying to be very careful with my money, and this car has served me very well. I've been all over the Fish Lake. All over those roads you took me on, Bob. Those are some rough roads, and this thing just keeps on going. Sometimes I have to have my graduate students get out because we bottom out on the backside. But this is what happens when you go to check something out and leave your camera in the car. Your graduate students go crazy and start taking pictures like this for you. I'd like to especially thank Bob Campbell and the Fishlake crew. I started last year, and Bob has given me an education in Aspen ecology on the Fishlake second to none. I've been able to spend 2 weeks with Bob and Dale looking at interesting patterns in the landscape so that we could address some of the physiological mechanisms that describe what's going on. And I'd like to thank Mary Lou for hosting me down in northern Arizona. We're going to try to continue that work. John Sperry, University of Utah, has done very good at hydraulic trace and training me on those methods. Rick for getting me started with the defense chemistry and being someone I could go to get advice on that. And then BYU and Charles Redd Foundation for support. And my good friend Ron Ryle, who's a mentor and a friend, and my thoughts are with him. Thanks. I powered through that. We could have a lot of discussion. Or not. Yeah.
Getting back to the drought effects on Aspen, you focused on cavitation as a mechanism of injury or stress. And I wonder what you think about the kind of subacute, in other words, if they close their stomates before cavitation occurs, but they have to keep them closed so long, for maybe years in succession of drought, that they are not able to photosynthesize and just run out of food.
Yeah, that's the dilemma. Okay? So, and it's that trade-off that the plants have to deal with. And it's interesting because John Sperry actually has shown that current year xylem production is dependent on previous year's photosynthesis rates. Okay? So yeah, if you close the stomates to limit cavitation, and that's carbon you're not gaining that could then be allocated to more xylem production in the current year that would be really helpful. So again, there is a dilemma there. And
which choice are they making, and under what conditions do they decide one way or the other?
Well, we know Aspen, there's a lot of clonal variation in Aspen. And so with that clonal variation, we can then go and test some of these ideas. Which strategy is better under a given situation, and kind of get at those questions. So we're really interested in physiological and morphological traits that allow a certain clone to be successful under these different conditions. So we're really at the beginning stages of being able to do this.
Any more questions? Let's give Sam another round of applause.